Hello students, I am your educator Dr. Hina Devnani and today we are going to study chapter number 8 of 11th standard, Cell, the Unit of Life. Here we are going to deal with the third part of this chapter. Two parts have already been discussed in which we have studied the introduction of cell theory, prokaryotic cell, the structure of the bacterial cell, eukaryotic cell and some organelles of eukaryotic cell like cell wall, cell membrane, mitochondria and chloroplast. Now today we are going to discuss with the other organelles of the cell. Have a look at these diagrams. We have already discussed these in our second part. This is the structure of an animal cell and this is the structure of a plant cell. Both are eukaryotic cells. We have already studied mitochondria. Mitochondria is present in both. See here, in plants are also mitochondria is there. And we have already studied chloroplast, cell wall as well as cell membrane. Now today we will be studying endoplasmic reticulum. Just have a look here, rough endoplasmic reticulum and this is smooth endoplasmic reticulum. So we are going to study this. We are going to study Golgi apparatus. You can see here Golgi apparatus. We are going to study about lysosomes, ribosomes and all the other extra organelles of a cell. First of all, let's study endoplasmic reticulum. Reticulum means a network. Endoplasmic means inside the plasma or cytoplasm. So the network that is present inside the cytoplasm is called as endoplasmic reticulum. See, a definition is given. A network or reticulum of tiny tubular structures scattered in the cytoplasm is called as endoplasmic reticulum. And in short, we are going to write it as ER. Here, ER divides the intracellular space into two distinct compartments. We have seen the structure of ER. It is present, actually it is continuous with the nuclear membrane. So it divides the cytoplasm or the intercellular space into two compartments. First is inside ER, that is luminal, and outside ER, that is extraluminal. ER is also divided into two types that we have already seen in the diagram, smooth ER and rough ER. Why this ER is called as smooth ER and why this is called as rough ER? Because on rough ER, ribosomes are present. Small bodies, small cell organelles, ribosomes are present on rough ER. And so the appearance of this ER in the microscope appears a bit rough. And when we look smooth ER through the microscope, its surface looks smooth. That's why the name has given from its appearance. Here there are no ribosomes, so they appear smooth. Here there are ribosomes, so they appear rough. Smooth ER and rough ER both are different in function. Smooth ER is responsible for synthesis of lipids, whereas rough ER is responsible for synthesis of proteins. Here you can see this is the major site for synthesis of lipids and this is involved in protein synthesis and the secretion of proteins. In animal cells, lipid-like steroidal hormones are also synthesized in smooth ER. Rough ER is extensive and it is continuous with the outer membrane of the cell. Okay, now here this is smooth endoplasmic reticulum, so we write it as SER. And this is rough endoplasmic reticulum, so we write it as RER. Here you can see the structure of endoplasmic reticulum. See, this is nucleus. As I said, that endoplasmic reticulum is continuous with the nuclear membrane. You can see this is the nuclear membrane. And here you can see that it is continuous. The endoplasmic reticulum is continuous with nuclear membrane. Okay, on this endoplasmic reticulum, you can see these rounded bodies. These rounded bodies are ribosomes. So if ribosomes are present, this ER can be called as rough endoplasmic reticulum or RER. See behind here there are no ribosomes, no rounded bodies. So this is smooth endoplasmic reticulum. So we can say that endoplasmic reticulum are divided into two parts based on the presence or absence of ribosomes. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Next organelle is Golgi apparatus. Golgi apparatus was discovered by Camillo Golgi. So the name has come from here, Camillo Golgi. Golgi apparatus consists of many flat, disc-shaped sacs or cisternae which are stacked parallelly. That means In a parallel manner, they are stacked one above the other. Here you can see in the diagram, see here. This is one cisternae, this is another cisternae, here third cisternae. They all are parallelly stacked with each other. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex phase and a concave phase. The convex phase is also called as cis phase or the forming phase, and the concave phase is also called as trans phase or the maturing phase. I show you how. Look here. Suppose if here there is nucleus, and the Golgi apparatus or the cisternae of Golgi apparatus which are near the nucleus consist of cis phase, which is also called as forming phase. That means what will happen here? The ER will produce proteins, and these proteins will be transported from this side, from the cis side. And when proteins are coming here, they are not completely mature. They are not properly made. They are not properly packaged. So they will come from here. This phase is called as forming phase. From here, the proteins will mature and they will come outside from this side. They're coming outside from this side when they are completely packaged and completely mature. That's why this is called as mature phase or trans phase. So we call this as cis phase and this as trans phase. This is also known as convex phase and this is known as concave phase. Cis phase, forming phase, and concave phase or trans phase as maturing phase. So Golgi apparatus principally performs the function of packaging material. As I said just now, that endoplasmic reticulum will produce proteins, lipids, and other materials, and these materials will not be in proper packaged manner before they enter the Golgi apparatus. When they enter the Golgi apparatus, their complete packaging is done over here. Golgi apparatus remains in close association with endoplasmic reticulum as materials to be packaged in the form of vesicles from the ER fuse with the cis phase of Golgi apparatus and move towards the maturing phase. I've explained this just now. Golgi apparatus and endoplasmic reticulum have a close connection. Why they have a close connection? Because endoplasmic reticulum is going to synthesize materials and these materials will be transferred to Golgi apparatus for packaging. Packaging, dispatching, distributing, everything is done in the Golgi apparatus. So the material will enter from the cis phase and they will move out from the maturing phase, that is trans phase. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of Golgi apparatus before they are released from its trans phase. This also I have told just now that mostly proteins will be synthesized by rough ER, and these proteins are not in a proper manner, not in a complete mature manner. So they will be sent to Golgi apparatus so that complete packaging is done. They are allowed to have a mature structure, and then these are released from the trans side of the Golgi apparatus. Golgi apparatus is the site of formation of glycoproteins and glycolipids. Glucose, proteins, lipids, everything will be formed in endoplasmic reticulum. But they are coming here for getting matured, for forming a proper structure. So Golgi apparatus will combine glucose and proteins to form glycoproteins. It will also combine glucose and lipids to form glycolipids. This is the structure of a Golgi apparatus. This phase is the cis phase and this phase is the trans phase. So from here they will enter. See, you can see here transport vehicles are there. These transport vehicles actually bring the materials from ER and give it to the Golgi apparatus. The Golgi apparatus will receive it and it will come here. Here you can see new forming vesicles. These rounded structures. Then here this is the trans phase. It is also known as shipping side of the Golgi apparatus. If you have little bit knowledge about factory, working of factory and all, you might be knowing that there is a production site and then there is a dispatch and then there is a shipping site, right? In production site, the materials will be produced, synthesized, and then they will be sent to the dispatch area. And here in dispatch area, it will be packaged properly in the suitable packing and all will be done, and then they will be sent to the shipping area, and from shipping area they will be distributed to various other factories or shops or whatever. Same working is done here also. So this is also known as shipping side of the Golgi apparatus. These are the secretory vesicles from trans phase, and these are also transport vehicles. So this is all about Golgi apparatus. Now, from the packaging process of Golgi apparatus, some membrane-bound vesicular structures are also formed, which are called as lysosomes. See, this is the structure of lysosome. It is a single membrane-bound structure. These are the membrane-bound vesicular structures which are formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzyme. Here inside, the lumen of lysosome consists of hydrolytic enzyme mixture. Hydrolytic enzyme means enzymes are known as biocatalysts. We all know what are catalysts. Catalysts are the chemicals which help in increasing the rate of reaction. Same thing enzymes also do, but enzymes do it inside living organisms. That's why enzymes are called as biocatalysts. They carry out the reaction, they actually increase the rate of reactions. So these are enzymes, and in our body, most of the functioning or almost all of the metabolic reactions are carried out with the help of enzymes. Now here it is written they are hydrolytic enzymes. Hydrolytic. Lytic means breaking and hydro means water. So the enzymes which are involved in breaking of substances in the presence of water are called as hydrolytic enzymes. Please note down this definition. The enzymes which are responsible for breaking down of materials in the presence of water are called as hydrolytic enzymes. And the enzymes that are present inside the lysosomes are generally lipases, proteases, and carbohydrases. Lipases means substances or the enzymes which break down lipids. Lipase stands for lipids. Proteases, that means enzymes which carry out the hydrolytic action of proteins, breaking down of proteins. And carbohydrases are responsible for breaking down of carbohydrates. And these work optimally at the acidic pH. Acidic pH means pH which is less than 7. More than 7 is called as basic pH, 7 is called as neutral pH, and less than 7 is called as acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. That means these enzymes are able to digest the whole cell. You might have studied in lower standards that lysosomes are also called as suicidal bags. Why they are called as suicidal bags? Because suppose if a cell, any cell, is in danger. Danger means either it has been attacked by a virus or bacteria, or there is some genetic mutation inside it, genes are misplaced, or there is some problem in the genes. And if this cell will survive, it will create a harm to the tissue or organ. So what the cell does here, in such situation or in such conditions, the wall of lysosome, this layer, membrane of lysosome is broken down and all the hydrolytic enzymes are released in the cytoplasm. Now here all the enzymes are present which have the capability to digest carbohydrates, proteins, lipids, nucleic acids. So what these enzymes will do, they will digest everything inside the cell and ultimately the cell will die. So actually what the cell has done, it has done suicide. It has killed itself. And for doing suicide, it has used lysosome. That's why lysosomes are also called as suicidal bags. And these lysosomes are very important because they are responsible for protecting the whole organism. Next organelles are vacuoles. Vacuoles are membrane-bound space which are found in cytoplasm. They are simply space, or we can say they are storage house for storing water, for any sap, fluid, excretory product, or any other material which is not useful for the cell at that time. And they will be stored inside vacuoles. The membrane of vacuole is known as tonoplast. Now, when we talk about plant cells, we know that vacuoles are very large in plant cells, whereas in animals, either they are absent or if they are present in a very small size. In plants, vacuoles are of great importance because they maintain the osmotic pressure inside the plant. In plants, the tonoplast means the membrane of vacuole, facilitates the transport of a number of ions and other materials against concentration gradient. Remember this against concentration gradient, active transport. I've explained you about passive transport and active transport. So here which transportation will be there? Active transport. So what the tonoplast will do, it will facilitate the transport of number of ions and other materials against concentration gradients inside the vacuum. So it will bring all the ions and other materials inside the vacuum. Even though inside the vacuum the concentration of those ions and those materials is very high, but still it will bring those materials inside. So what it is doing, it is carrying out active transport. Now, if we talk about animals, in animals, vacuoles are present either in the form of contractile vacuoles like amoeba or in the form of food vacuoles. Food vacuoles are also present in amoeba. What this contractile vacuole will do, it is responsible for excretion. And what is the function of food vacuoles? They are responsible for digestion of food. And these food vacuoles are formed by engulfing the food particles. In 10th standard, we have already studied how the amoeba occupies the food. What it does, it first withdraws its pseudopodia and then it covers the food material, and then it will take it inside. It will ingest it, engulf it, and then a food vacuole will be formed in which digestion, absorption, assimilation processes will be going on. So this was about vacuole. Next organelle is ribosome. Ribosome was also present in prokaryotic cell. Ribosomes were first observed under electron microscope, a well-developed microscope, not with our common laboratory microscope, not like our common laboratory microscope. It was first observed by George Palade. Ribosomes are composed of RNA and proteins. Ribosomes are not bounded by any membrane, and that's why they are present in prokaryotic cells also. In prokaryotic cells, we have studied that ribosomes are 70S, but in eukaryotic cells, these are 80S. And how this 80S is formed? It is formed by combination of 60S and 40S. Please remember this, 60 and 40 will not make 100 over here. And remember 30 and 50 were there in prokaryotic. 30 and 50 will make 70S, 60 and 40 will make 80S. What is this S? We are saying it again and again. S is the sedimentation coefficient. That is, it determines the sedimentation rate. S can also be denoted as Swedberg's constant or Swedberg's unit. So you will find in some books that S is Swedberg's unit, and in some books it will be written as sedimentation coefficient. Both are correct. Moving further, next organelle is cytoskeleton. Cyto means cell. Skeleton of the cell, that is known as cytoskeleton. It is an elaborate network of filamentous proteinaceous structure which are present inside the cytoplasm. Now, what is the function of skeleton in our body? Bones. It gives us support. It is responsible for our movement. It is also responsible for maintaining the posture of our body. Same thing the cytoskeleton will do in cell. The function of cytoskeleton in cell, functions of cytoskeleton in cell are mechanical support. It provides mechanical support to the cell. They are responsible for motility of the cell. In amoeba, that pseudopodia formation and all is carried out with the help of cytoskeleton. It is also responsible for maintaining the shape of the cell. See here you can see the structure. These are the structures or the fibers of cytoskeleton. They can consist of actin, myosin, tubulin, and different fibers will be there. So this is just a structure of cytoskeleton inside the cell. They are made up of proteinaceous fibers, actin, myosin, tubulin. Remember these three names. Next two organelles are cilia and flagella. Cilia and flagella, both are responsible for locomotion and movement of cell. Cilia are small structures, very small hair-like structures, and they are present on the entire cell. Whereas flagella is tail-like structure, and flagella are generally found on one or the other ends of the cell. So these both are different in external structure, but their function is same and their basic structure is also same. We can see here they are hair-like outgrowths of the cell membrane responsible for locomotion and movement of cell. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. In paramecium, what happens? Suppose if food particle is present in the surrounding fluid, water, We all know that paramecium has cilia all over its body surface. So what the cilia will do? They will start movement, and because of that movement, the water will change its current. Water current will change. That means it will work like an oar over here. So what will happen here? Because the water current is changed, the food particle will flow along with the water and it will come near the paramecium so that paramecium can consume it. Flagella are comparatively longer. Example of flagellated organism is Euglena. It has tail-like structure which is known as flagella. It is also present in human sperm cell. The sperm has a tail-like structure, that is a flagella. Eukaryotic cilium or flagellum are covered with plasma membrane. Their core is called as exoneme. They possess a number of microtubules running parallel to the long axis. The exoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Now this paragraph will be studied by seeing the diagram. You can see the diagram over here. See, this is Euglena. It has a tail-like structure which is a flagellum. This is a paramecium. It has hair-like structures which are called as cilia. This is the basic structure of a cilium and a flagellum. Here in the center, it will have axis, central axis. You can see here what is written. That core is called as exoneme, and it possesses a number of microtubules running parallel to the long axis. So this will be the axis, and here there are many microtubules present here. When we see the structure in a microscope, we'll find that in the center there are 2 complete rings, whereas in the periphery there are 9 doublets. Doublets means 2 rings together, in which one ring is complete, one is incomplete. Just count it, it will be 9. See, from here to here when we see, these are 9 doublets, and in that 9 doublets also one is complete, one is incomplete. The center here there are 2 rings. So what we call this type of structure, because here there are 9 and here there are 2, so we call it as 9 plus 2 arrangement. You can see here it is written 9 plus 2. This is the 9 plus 2 arrangement. Clear? Now, cilia and flagella are also covered by a plasma membrane. Here this is the covering, plasma membrane. In the center we have central microtubules. Central microtubules and these doublets are connected by some spoke-like structures which are called as radial spokes. So you can see these are radial spokes. Now these doublets are interconnected with each other. They are interconnected with each other by the help of nexin. And here you can see some arms coming out from these doublets. These arms are called as Dynein arms. The complete ring is called as A tubule. The incomplete ring is called as B tubule. So it is a doublet microtubule. This structure is very, very important. The diagram is very important. It is asked in many exams, as well as the theory part of this structure. Central, there are microtubules, 2 microtubules which are complete rings. Surrounding in the periphery, there are 9 doublet microtubules. Out of these, the A1 is complete and the B1 is incomplete. They are connected with each other with the help of nexin. Doublets are connected to the central microtubules with the help of radial spokes, and they all are surrounded by plasma membrane. The structure is very important. Next, we'll be talking about centrosome and centriole. Centrosome is an organelle usually containing 2 perpendicular lying centrioles surrounded by amorphous pericentriolar materials. Have a look. When we talk about this entire structure, this is called as centrosome, and centrosome consists of 2 perpendicular lying centrioles. Centriole has an organization like the cartwheel. Similarly, like cilia and flagella, these are also made up of 9 evenly spaced microtubules. In cilia and flagella, the microtubules were doublet, but here the microtubules are triplet, and these microtubules are made up of tubulin. Central part of the centriole is also proteinaceous, that means made up of protein, and the central part is called as hub. It is connected with peripheral tubules by radial spokes. Same like cilia and flagella, radial spokes were there to connect the peripheral tubules to the central tubule. Here also radial spokes are present. The centrioles form the basal body of cilia or flagella. From these centriole only a basal body will arise. From basal body, cilia and flagella will arise. Centrioles are also responsible for forming spindle fibers, which actually give rise to spindle apparatus, which are important during cell division. But these centrioles are present only in animal cells and not in plant cells. You can see here, inside there will be hub, and here you can see 9 triplets are formed. See, 3 tubules or 3 tubes are together. They are called as triplets. And these both, this is one centriole from here to here, and this is second centriole. They both are arranged at 90 degree. You can see if they are arranged perpendicularly to each other. These are very important during cell division. Apart from all these important organelles, there are some microbodies which are present inside the cell. What are these microbodies? They are small membrane bound vesicles, and they contain various enzymes which are responsible for carrying out metabolic reactions inside the cell. Microbodies are present in both plant cells as well as animal cells. Now we'll be moving with the main part of a cell, that is nucleus. Nucleus is also called as the brain of the cell because all the messages are given by the nucleus. Like how brain gives messages to an organism, nucleus is responsible for carrying out all the functions in a cell. Nucleus was first described by Robert Brown. The material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. What Fleming did, he studied the cell in a microscope with the help of some stains, basic stains. And when he put or gave these basic stains to the cell, he found that inside the nucleus something colored was visible. That means something was visible which had already taken up the dye. So he named it as chromatin. The interphase nucleus has nucleoprotein fibers which are called as chromatin. This chromatin material is present in the form of fibers, thread-like structures, when a very thin thread-like structure. Interphase nucleus means the nucleus which has not entered the cell division yet. It is going to enter. Okay, that is known as interphase nucleus. Interphase nucleus also consists of nuclear matrix and one or more spherical bodies which are called as nucleolus. So three things are present inside the nucleus: chromatin, nuclear matrix, and nucleolus. When we talk about the envelope or the membrane of nucleus, we see that it is double membrane bound. Double membrane bound means it has one outer membrane and one inner membrane. Now between these two membranes, there is a space which is called as perinuclear space, which is filled with a fluid which is known as perinuclear fluid. Outer membrane is generally continuous with endoplasmic reticulum, that we have seen already. When we were studying about endoplasmic reticulum, we have seen that it is continuous with the membrane of nucleus. So outer membrane will be continuous with endoplasmic reticulum. Now endoplasmic reticulum has ribosomes on it, so the outer membrane of the nucleus will also bear at some places ribosomes on it. At a number of places, this membrane, outer membrane and inner membrane both, they are interrupted by minute pores. Small pores are there, and these pores are called as nuclear pores. What is the function of these pores? These pores provide passages or channels so that RNA and proteins from inside can move to outside, or some material which are present outside, if it is required by the nucleus, can come inside the nucleus. Normally there will be only one nucleus per cell, but in some cells the number of nuclei can increase, and some cells may even lack nucleus, such as erythrocytes of mammals and sieve tube cells of vascular plants. The nucleoli are generally spherical structures, rounded bodies, and they are present in nucleoplasm. Nucleoplasm means the fluid present inside the nucleus. These nucleoli are non-membrane bound. It is not having any membrane, and it is a site for ribosomal RNA, that is rRNA synthesis. What is this rRNA that we'll be doing when we are studying or when we are dealing with biomolecule chapter? So here, for the synthesis of rRNA, these nucleoli are present. During cell division, what happens, as I told earlier, that before the nucleus or the cell enters in division phase, the genetic material is in the form of a network-like structure which is called as chromatin. But during cell division, what happens, this chromatin network gets condensed. And when it gets condensed, it has a proper shape. A shape with a rounded center and having 4 arms, which is called as chromosome. So chromatin network condenses into chromosomes. These chromatin threads or chromosomes contain our DNA, that is the genetic material. So we call chromatin as genetic material. And they also contain some proteins along with it. Now, why proteins are present here? Because DNA is negatively charged. So, to keep it neutral and to keep it stable, proteins are present. Basic proteins are present, which are positive in charge and they are basic. DNA is acidic. So, here basic proteins are present. These basic proteins are called as histone proteins and non-histone proteins. Along with DNA and proteins, even RNA is present in chromosomes. Now, every chromosome essentially has a primer constriction that I said just now. In the center, it will be rounded structure and it is called as centromere. Surrounding the centromere on both the sides, there will be disc-shaped structures, and these disc-shaped structures are called as kinetochores. What is the function of these kinetochores? They are responsible for attaching to the spindle fibers. During cell division, what happens? The chromosome is split into two parts and half will be divided in both the cells. So for splitting it, spindle fibers are produced on both the sides, two polar sides of the cell. In detail, we'll be doing when we are studying 10th chapter. For now, just have this basic information. What will happen here? Spindle fibers will be present on the two corners of cell, and from centrioles will be present, and from there they will produce spindle fibers. These spindle fibers will be attached to kinetochores, and these spindle fibers after attachment will shrink. Because of their shrinking process, what will happen? The centromere or the chromosome will be split into two parts. So that is the function of kinetochores. They are there for attachment with the spindle fibers during cell division. This is the structure of a nucleus. You can see a rounded body. Inside there is nucleolus. This is chromatin network, thread-like structure. This is nuclear envelope, inner membrane, and here there is this outer membrane. This blue one is the inner membrane, this purple one is the outer membrane. And here you can see that it is continuous with rough endoplasmic reticulum. Now on this envelope, pores are present. You can see here, these are nucleopores, responsible for transportation of substances from outside to inside and inside to outside. So this is the basic structure of a nucleus. Now the main part of the nucleus, chromosomes. Chromosomes are divided into two parts, P and Q arms. First have a look at the diagram. See, this is a typical chromosome. It has four arms, two above and two below. And here there is a rounded body. It is called as centromere. So here what will happen? These two are known as P arm and these two are known as Q arm. The centromeres can be located in different positions and this forms the basis for four different classes of chromosomes. Now, based on the position or location of centromere, there are four different classes of chromosomes. Metacentric, submetacentric, acrocentric, and telocentric. In metacentric, what happens? The centromere is in the middle. See here, you can see the centromere is present exactly in middle. That means P arms and Q arms will be same, comparable length. They will be of same length. Metacentric chromosomes are chromosome number 1, 3, 16, 19 and 20. First of all, let me tell you how many chromosomes are present in a cell. 23 pairs of chromosomes are present in our cell. 22 pairs are autosomes and the last pair is the sex chromosome. So from 1 to 22, we denote it by numbers, normal Arabic numbers. And the 23rd pair is denoted by either X or Y. It is a pair, so either it can be XX or it can be XY. So that we have seen about metacentric. Submetacentric, here what will happen? Centromere is off center, it is not exactly in the center. The P arm will be shorter as compared to Q arm. And it is present in which chromosome? Chromosome number 2, chromosome number 4, 5, 6, 7, 8, 9, 10, 11, 12, 17, 18 and chromosome number X. See here, submetacentric. P arm is shorter and Q arm is longer. Third type is acrocentric, in which centromere severely offset from the center, leading to much shorter P arm. Chromosome number 13, 14, 15, 21, 22, and Y chromosome. See here. This is acrocentric. P arm is very short. And last, telocentric. Chromosomes are found at the centromeres are found at the end of chromosome, meaning there is no P arm, and these chromosomes are not found in humans. You can see the structure. Now, just now I told you that during cell division, the centromere will divide and it will be split into two parts. You can see here. These are two dividing cells, and the centromeres have been split, and half part has gone here, half part has gone here. So this type of structure will be there. One P arm, one Q arm. Here also one P arm, one Q arm. In metacentric, it is very difficult to identify which one is P arm and which one is Q arm. But here in submetacentric, you can see this is P arm, this is Q arm. Here again, this is P arm and this is Q arm. In acrocentric also, you can identify P arm and Q arm, and here in telocentric. This is the structure of a typical chromosome, a well-developed condensed chromosome. Sometimes what happens, some chromosomes have non-staining secondary constriction. Centromere is called as primary constriction. Sometimes secondary constrictions are also present. You can see here, see this one and this one. At proper location, constant location will be there. Now what will happen when these constrictions are there, it will give the appearance like a satellite. You can see this is looking as if it is a satellite of this chromosome. So this type of arrangement is called as satellite arrangement. These are the secondary constrictions. So that was all about our 8th chapter. Today we have completed our 8th chapter. We have already dealt with prokaryotic cells and eukaryotic cells, all the organelles of eukaryotic cell. Once again, I want to remind this is a very important chapter, and that's why I've taken it so soon. It is 8th chapter, but still we are studying so early because this is a very important chapter. All the organelles are very important. All the diagrams are very important. So start studying from now. Prepare well for all your exams as well as competitive exams. If you have any doubt, you can contact me. Till then, take care and stay safe.